and let's look at the effect of temperature on enzymes. So what I draw here is a temperature versus rate of reaction graph. So what it shows here, we've got an optimal temperature. You can just see here we've got this peak of the graph, and what I do is change this to about 37. Our optimal temperature in most enzymes within our body will be about 37 degrees. At cold temperatures, as the temperature keeps rising, the temperature is directly proportional to the rate of reaction, or we can call it a linear relationship because it's not always directly proportional. After this optimal temperature, we see enzymes being denatured and the rate of reaction then lowers. The effect of temperature depends on what enzyme we have. So some enzymes prefer cold temperatures. So this green graph shows an optimal temperature here, and we'll say pretend that's about 30 or 25. Okay, they have a lower optimal temperature. This one was the original one, saying we preferred about 37, and then we've got this one, which could be an example like thermophilic bacteria, which prefer really high temperatures. If we look at Taq DNA polymerase, which I'll use as an example later, from the PCR reaction, it lives in naturally occurs around volcanoes, this bacteria, and it's used to really high temperatures. So its optimum temperature in our PCR reaction is 72 degrees, which is absolutely huge. These two equations, rate of reaction equals 1 divided by time to reach the end point. To work out the temperature coefficient, we do T plus 10 divided by T, and both the answers in degrees C. So what this equation basically means is, so if we want to work out between 25 and 35, T plus 10 is 35, so 108 divided by T, which is 50. So basically the larger temperature, so 35 degrees minus 10 is our T, and we put that at the bottom. It's quite a confusing equation. Maybe there's a better way of explaining it, but I just remember that if you've got two temperature values, one's 10 degrees above the other, you want to use the higher one first and divide it by the lower temperature value. So 108 divided by 50, and then you get 2.16. And another example, if we just redo it to make this clear, between 35 and 45, you put the 45 value on the top, so 100 divided by the 35 value, which is 108, and you get a Q10 of 0.98. You might want to do a bit of further reading into that, because I'm not the best mathematician in the world. Probably didn't explain it that well. So I've spoken to you about this graph and saying it rises here, it reaches an optimum, and then it falls. I've not really explained why that happens yet, so this is what it's doing here. So in cold temperatures we've got low kinetic energy, so molecules are moving slowly, we have less successful collisions, and therefore less enzyme substrate complexes form. Less EPCs, so less enzyme product complexes, because if we've got low enzyme substrate complexes, then we can have low enzyme product complexes, therefore we get less product and a lowered rate of reaction. So as the temperature rises, our kinetic energy rises, we get more successful collisions, and our rate of reaction increases. But we can get too hot. So this is the opposite in hot temperatures. I won't speak you through that, it speaks for itself, until a certain point. So we reach our optimum temperature, everything's been going really well, and beyond this everything starts to go down the drain. So hydrogen bonds in tertiary structures start to break because they're vibrating so much under this high temperature. More bonds break as the temperature increases. The active site no longer is complementary. ES complexes no longer form, therefore we get less enzyme product complexes, less product, and our enzyme becomes denatured. And once again, we can have different optimum temperatures, because remember we're dealing with proteins here. Some proteins have different tertiary structures. Some proteins have a different primary structure. Well, all proteins do, different proteins, because they've got different amino acids within their sequence. So if we've got this protein cysteine, which contains sulfur in the R group, we've got lots of disulfide bridges or links, and therefore we're more resistant to heat, high temperatures. And our example was Taq DNA polymerase, has lots of cysteine amino acids, and it can withstand higher heats than normal temperatures. So the amount that enzyme can withstand heat or withstand low temperatures depends on both its primary and tertiary structure, because obviously the primary structure decides the tertiary structure. But if we've got this cysteine, we can get disulfide bridges in our tertiary structure, and then we become more resistant to heat. So temperature is quite straightforward to understand. All these things are written here are really key words, and always write stuff like low kinetic energy. Successful collisions is a huge one. Always think about collisions and whether they're successful or less successful. ES complexes. So now let's look at pH. So pH is something else that affects the enzyme activity. So we can have 0 to 6, which is acidic, 7 is neutral, and 8 to 14 is alkaline. So hydrochloric acid and sulfuric acid dissociate into protons and negatively charged ions. So hydrochloric acid can form positive and negative ions. These positive ions end up, hydrogen ions make acids. H2SO4 forms H plus and HSO4 minus. Lactic acid dissociates into H plus and lactate. Pyruvic acid dissociates into H plus and pyruvate. So you notice a recurring theme. This H plus creates this acidity in all of our different acids. Buffers can resist changes in pH. So these chemicals can donate or accept hydrogen ions. Proteins such as hemoglobin can also donate and accept protons, and so act as buffers. So as we know, hemoglobin can resist changes in pH, as it can accept and donate protons. And there is another video that I've done, I can't remember the unit number, but it does tell you about how hemoglobin acts as a buffer. So how do changes in pH affect? So hydrogen ions have a positive charge, they're protons. They're attracted towards negatively charged ions, molecules and parts of molecules. So hydrogen bonds and ionic forces between amino acids hold the protein's tertiary structures, you should know, particularly at the active site. So those bonds are really important for holding the structure of the active site. If we have excess hydrogen bonds, then let's just get this text back to something more relevant. If we've got excess hydrogen bonds, then this interferes with the ionic forces, so the active site of the molecule will change shape. The rate of reaction is lowered as the substrate molecule no longer fits well into the active site. So increased hydrogen ions alter the charges of the active site as more protons gather around the negatively charged groups, interfering with the binding of the molecule to the active site. So basically we're just interfering with all these forces of attraction. The active site basically deforms, and the same happens with high pHs. Okay, so if we have negative atoms interacting with our ionic forces, etc., then the same thing will happen in high pHs. But mostly OCR wants to teach you about the effects of the lower pHs. but the effect is equal at both ends of the scale. So let's look at this graph comparing different types of enzyme and their effect, how they're affected by protein. So what we get here is a bell-shaped curve. So I've drawn on three here. We've got pepsin in red, amylase in green, and trypsin in purple. So pepsin is used to stomach. It works in the stomach acid between 1 to 2 pH. So we see a really optimal pH. It probably should. The graph should shift along a bit further. Don't use my numbers on this graph. It's really quite rough. But we see the optimum should be about 1 to 2 to 3. Okay, because that's where the enzyme works. Amylase is extracellular. It prefers a neutral pH of 7. It works in the mouth. And trypsin, it's got this bell-shaped curve again, but it works best at 7.8 pH, as that works in the alkali bile used to neutralise stomach acid in the small intestine. So we can see enzymes can have a huge range of optimal pHs, and it is all to do with their tertiary structure and their primary structure, which amino acids they're formed of. And that is literally all you need to know about the effect of pH on substances. For each one of these, you want to know the shape of the curve, so learn the one for temperature, learn the one for pH, and now we're going to look at the effect of changing the concentration of substrates. As more substrates are added, the rate of reaction increases as we can form more ES complexes, more products are formed. So our substrate concentration is the limiting factor on the rate of reaction. They do not have to increase the rate of reaction at a certain point when it increases, when it reaches maximum rate. So all enzymes become occupied at this maximum rate. Anything below our maximum rate of reaction, the substrate concentration is our limiting factor. This graph, which will help explain what I've spoken about. So substrate concentration versus rate of reaction, and this point here drawn in orange is our max rate of reaction. So up to our maximal rate of reaction, substrate concentration in this area of the box is the limiting factor. Beyond that, substrate concentration is not the limiting factor, it's something else, and usually it is enzyme concentration. Let's just think about it. If you've got loads of substrates, then you get loads of successful collisions, loads of ES complexes form, and then you have basically a surplus of substrate, and it doesn't affect the rate of reaction. Okay, so because if all the ES complexes are full, then the thing that becomes the limiting factor is your enzyme concentration. So enzyme concentration becomes the limiting factor at the maximal rate of reaction for the substrate. At this point, increase in substrate concentration will not, if the substrate concentration is limited, substrate concentration is the limiting factor. But if we've got a surplus of substrate, then we see that enzyme concentration becomes the limiting factor, and it's the same graph here. So at this point, the substrate becomes the limiting factor. So if we increase the enzyme concentration, we eventually reach a maximal rate of reaction. At that point, our substrate is the limiting factor. We need more substrate to keep this curve going upwards, which I'll draw on later. We're reducing surplus to requirement enzymes, eliminating abnormal harmful proteins. Okay, so we're constantly degrading enzymes, so quite often enzymes can be our limiting factor. We don't have enough. But what you've got to understand here is the real correlation between substrates and enzymes. Once the substrates reach their maximum rate of... So, okay, let's slowly speak through this graph. So we're increasing our substrate concentration. As we do that, the rate of reaction goes up. As we're forming more ES complexes, more EP complexes, etc. Okay, until this point here where we increase the substrate. We've got so much substrate that enzymes are not capable of dealing with it at the same time. Then we reach our maximal rate of reaction with that concentration of enzymes. Therefore, enzymes are our limiting factor. So if we increase our enzyme concentration with a fixed amount of substrate in this graph, we then reach the same plateau where substrate becomes the limiting factor is we've got loads of enzymes and some of them got free active sites not being able to bind to anything because there's not enough substrate. So substrate is our limiting factor. These two graphs are the same and they are basically inversely proportional to one another, enzymes and substrates. And that is the line that you'd see if there was more substrate. It would keep going up. So now let's look at initial rate of reaction. So when we first mix our enzyme and substrate, we see our fastest rate of reaction. And you might be thinking, oh, that's not really represented on this curve. And I'm going to change something on these axes in a second. But first of all, let me just explain to you. So when we first mix them, we've got lots of free enzymes with free active sites for lots of ES complexes to form. So the rate of reaction is very high at the very start of the reaction. At the initial rate of reaction is the highest. Okay, this is because we've got these free active sites, lots of substrates ready to be reacted with, so we get that high rate of reaction. If the reaction proceeds, more product molecules are produced, therefore the substrate concentration decreases and we get a lowered rate of reaction, and that happens instantaneously. The rate of reaction decreases from the start, but the start of the reaction is where we've got the highest rate of reaction. This is the important bit in black. Thus, initial rate of reaction gives maximum rate of reaction for an enzyme under a particular experimental situation. So that's basically saying the start of the reaction is where we see the rate of reaction being fastest. So all of these graphs were originally rate of reaction. They should be initial rate of reaction. So the initial rate of reaction is what's being recorded. So the amount of reaction we get when we first add the substrates and enzymes together, and that's the points we record on these graphs. And you basically keep doing the reaction. And the same here, initial rate of reaction. In the book, the pH one was still rate of reaction. Okay, don't know if that was a mistake, but I'm just copying exactly from the book. Don't think you should be too concerned about it though. So to work out your initial rate of reaction, use this A over B. So B basically equals 1 second. So how far you go along in 1 second, that's basically your distance of your distance of B, how long the second is on your graph. So you do 1 second and that's B, and then you go up and that is A. So A is basically the amount of increase in rate of action you get in 1 second. So A over B finds out your initial rate of reaction. Now let's have a look at competitive inhibitors. So inhibitors in general, competitive inhibitors being the first one. So competitive inhibitors are something that binds to the active site of the enzyme. They reduce the activity of the enzyme just like all inhibitors do, but basically they act as a substrate when they're not actually the substrate the enzyme wants. So they bind to the active site, blocking the active site so no more ES complexes can form with that enzyme until it moves away. Obviously this is an issue, so if you have something blocking the active site, you can't form ES complexes, you can't form the enzyme product complexes, and you can't form the product in general. So big issue for enzymes, but one way it can be overcome is by increasing the concentration of substrates. So what I do is represent it here in a diagram. If we increase our substrate concentration, then we help dilute the solution, basically dilute the inhibitor solution. We have more chance of successfully colliding with a substrate rather than colliding with an inhibitor to form an enzyme inhibitor complex, which is a bad thing. So if we've got more of these pink substrates, we've more chance of forming ES complexes, successful ES complexes, and then we can overcome the issue of substrate. So the higher the substrate concentration, the lower the effect of our inhibitors. And that's pretty much what I write there. This here we call an enzyme inhibitor complex when the inhibitor forms in the active site, but it is a form of competitive inhibition, which is usually temporary. However, we can have irreversible competitive inhibition, and we call this inactivators. So anything that binds to the active site and will never be removed is an inactivator. Now we need to look at non-competitive inhibitors, and they are something that binds to something that's not the active site, so the allosteric site of the enzyme. They're potentially more harmful than competitive inhibitors, as they disrupt the enzyme's tertiary structure and change its shape, making the active site no longer complementary to the substrate, so no more ES complexes can form, and we get a significantly reduced rate of reaction. Okay, and that's pretty much all you need to know on non-competitive inhibitors. They bind to that allosteric site, and they just like with pH and temperature, they can denature enzymes. They destroy its tertiary structure. The active site's no longer complementary. We can't form those ES complexes, and we get that decreased rate of reaction. And that's me trying to represent what happened here. So that was our substrate. It fits perfectly in here. We get this non-competitive inhibitor. It binds to the allosteric region. We get a change in shape of the active site, and now this substrate can no longer fit in. We can no longer form that ES complex. So this is the normal rate of reaction for an enzyme when we're increasing substrate concentration, as you already know. This is what it will look like for a competitive inhibitor. So an increase in our substrate concentration will decrease the effect of our competitive inhibitor. Eventually we'll see an increased rate of reaction. And for our non-competitive inhibitor, increase in our substrate concentration has no effect on the rate of reaction, because the non-competitive inhibitors just destabilize so many enzymes, and we just get a maintained very low rate of reaction. So end product inhibition is when the product molecules may stay tightly bound to the enzyme, and usually they're a form of reversible non-competitive inhibition by product molecules. So our product molecules essentially act as an inhibitor for the enzyme itself, or an enzyme earlier in the metabolic process. And you may think that's pretty weird. Why would you want the product to inhibit the reaction? It's a bit backwards. But for metabolic reactions, we don't want to produce too much of our product. It's important to have some sort of method to stop the reaction from happening. This is how we do it. It's very clever. So this is a metabolic reaction. We've got this enzyme, this substrate, which is circle, then it produces this triangle product. This is obviously not how it will be in real life. I'm just making it easy to visualise. Then this triangle becomes a substrate for the next enzyme in the reaction, forming this square. This square is the substrate for the next enzyme in the reaction, and this forms a star, and this star inhibits the first enzyme in the reaction through non-competitive inhibition. And we call this end product inhibition. So this product prevents the metabolic cycle or the metabolic pathway from being maintained. So that is all you need to know on inhibition. Finally, let's look at some poisons and medicinal drugs and how they can inhibit certain enzymes. This is basically just some examples that OCR want you to know for the exam. So first of all, we've got cyanide, and it's a very famous poison. Potassium cyanide, KCN, is highly toxic because it inhibits aerobic respiration and also inhibits catalase. So when ingested, KCN is hydrolyzed to produce hydrogen cyanide, a very toxic gas which can readily dissociate into H+ and Cn- ions. And these Cn- ions bind irreversibly to an enzyme found in mitochondria and inhibit the final stage of aerobic respiration. So this inhibition of the final stage of aerobic respiration means the earlier stages cannot run and aerobic respiration stops, and therefore sort of like suffocates you from within. Then you've got snake venom, and snake venom of a green mamba snake contains a chemical that inhibits enzyme acetylcholinesterase, and we know that breaks down the acetylcholine at our neuromuscular synapses. So to break down the neurotransmitter acetylcholine, if the enzyme is inhibited, then we'll get constant contraction of our muscles. We will never be able to break down acetylcholine. Then we'll get paralysis. And if we get paralysis of muscles involved in breathing, then the victim will die of suffocation. Then we've got some medicinal drugs that act by enzyme inhibitors. So not poisons. These are things that help us. So aspirin is a drug that probably everyone here has taken. It prevents the formation of prostaglandins, that are cell signalling molecules produced by cell walls when tissues are affected or damaged. So this salicylic acid binds to enzymes that catalyze the formation of prostaglandins, and by preventing the formation of prostaglandins, we can prevent this nerve cells being sensitive to pain. So it decreases the sensitivity to pain, which is helpful for a lot of people in so many cases. Then we've got ATPase inhibitors. So extracts from purple foxglove leaves have been used for centuries to treat heart failure and atrial arrhythmia, which is an abnormal beat of the atria. The chemicals are now identified as cardiac glycosides, also known as digitalis, digitoxin, digitalin, or digoxin. They inhibit sodium-potassium pump in the cell membranes of heart muscles and allow more calcium ions to enter the cell. So calcium ions increase muscular contraction, and this strengthens the heartbeat. ACE inhibitors are medicinal drugs that inhibit the angiotensin converting enzyme ACE, which normally operates in the metabolic pathway that ultimately increases your blood pressure. So ACE inhibitors bring down blood pressure to lower blood pressure in patients with hypertension. They help treat heart failure. So low dose is given at first, and the patient's blood pressure is checked in case it falls too low. They minimise risk for second heart attack or stroke in patients who have suffered myocardial infarction. So ACE inhibitors inhibit angiotensin converting enzyme ACE, which increases your blood pressure. So by inhibiting that, we can help decrease your blood pressure. Protease inhibitors such as amprevanir and ritonavir are used to treat some viral infections. So protease inhibitors can help prevent replication of virus particles in host cells by increasing protease enzymes so that the viral coats cannot be made. These inhibitors often inhibit viral protease enzymes by competitive inhibition. So of course, competitive inhibition is not quite as effective as non-competitive inhibition. Most of these inhibitors are non-competitive, but these protease inhibitors use competitive inhibition. And the last one to learn is nucleoside reverse transcriptase inhibitors. So basically inhibits the action of viruses to manipulate DNA to replicate the viral DNA by using the viral DNA as the template. So it basically stops viruses from reproducing. That is all on Unit 2.4. Really long video. It's going to take a lot of editing. Thanks for watching.